Okay, so we had just started talking about handling and care of the microscope. Okay, remember that it is always carried with two hands, one on the base, one on the arm. Okay, and a couple of other things that we didn't get a chance to talk about. Once in a while, people, touch the lens or whatever, or stuff gets on it, it gets dirty, and it makes it difficult to see through. Okay, there is a proper cleaning procedure for the lenses. Proper cleaning procedure is not, okay, because I have seen people do that. Okay, it's disgusting. First off, do not lick your finger and smear it all over the lens. Okay, that's not going to make the situation better. Okay, also, you would not believe the amount of microscopic grit that is always on your skin, especially your hands. Okay, and if you do that, you could grind that microscopic grit into the lens and scratch it. Remember, the job of a lens is to make things look bigger. So even a microscopic scratch looks like a gaping chasm, okay, when you look through the microscope. So please avoid doing that. Okay? Other things, I see people take their shirt and they're thinking, yeah, here we go. Okay, same thing, especially if you're wearing something synthetic. Okay, synthetic fibers are often quite hard. Okay, and we don't want to be rubbing them onto something that cannot be scratched. Okay. So what we have in the lab is special lens paper for cleaning. Okay, you use lens paper once, right? So you take a small sheet of it, clean all of the lenses off, and throw it away. Whatever it picks up then doesn't end up back on a lens afterwards. Okay, they're disposable pieces of lens paper. So if you find that we're looking through here, you can see a big fingerprint on the lens or something like that, just come get a piece of lens paper and you can clean it off. Okay, that's the safest way to preserve the equipment. All right, questions on that? Okay, what we need to look at today is how do we show people what we have seen underneath the microscope and communicate to them how big it is, right? Well, we do that through a lab diagram. Now, some universities still would have you do these by hand, which I think is stupid, okay? The technology is now available to do a much better job of the way I had to draw lab drawings, which for a person of my artistic ability was awful, okay? I had to draw all kinds of stuff that I looked at under a microscope freehand, okay? I've never seen a stick paramecium until I drew one, okay? Yeah, it doesn't get you a good mark, right? So what we're going to do instead is we're going to use the phones on our cameras. The phone's on our cameras. The camera's on our phone, the other way around. The camera on your phone, okay, to take pictures through the microscope lens. It works pretty well. It takes you a little bit of time to kind of get the hang of getting it in the right position, but once you do, generally it takes a pretty decent picture of what you're looking at. Then we'll insert that picture into our lab report. Okay, same way we did with the planning for your toxin project, that kind of thing. So you'll take those pictures, send them to your Isidore email, or if you're using your phone directly, you can insert them directly into Google Docs from your phone. Either way will be fine. Okay, we'll do our best to do some labeling on those. Okay, so you may have to. For some people, it was in a matter of I take my picture, I print the picture, and then I draw it on, I take another picture, okay? And that's fine too, okay? That'll work, and sometimes that's a little bit easier, but we do want to use the picture we took because it's way better than the drawing we could ever render ourselves, okay? So things that need to be included, okay? Labels, title, okay? Name of the specimen, and it's not Bob, okay? It's like paramecium or amoeba, okay? It's not Bob the amoeba, okay? So you don't have to give it a real name, just tell me what it is, okay? The size of it, the magnification you viewed it under, okay, all of that stuff needs to be on a proper lab diagram. So there's a format for that, okay? And the proper format for a lab drawing looks like this, okay? So imagine that there's a rectangle here that represents a sheet of paper. Your name and the date go in the top right-hand corner, always, okay? Name and the date always go in the top right-hand corner. Then you have your drawing, or picture in this case, and you're going to make it as big as possible. Okay? We want it to take up as much of the page as humanly possible, so it's very big and easy to see all the parts. Okay? Your labels, notice all the labels go to the same side. That way you can shift your picture to one side and have room for all your labels on the other, and it doesn't end up looking busy. Okay? If you've got labels going everywhere in every which direction, it gets really, really busy looking. Right? The other thing is you'll notice that all of the lines are horizontal. Okay? They all go out horizontally from the structure. If you have two structures that are really close to each other, like let's say I needed to label this object right here, I can't draw straight over because then I'd overlap the uterus of this flatworm here. Okay, so what I would do is go up and then over. Okay, always ends in a horizontal line to label the structures. You're probably not going to have too much of an issue with that because we're not going to label too many things, especially on this first lab where we don't really know the parts of the cell yet. Okay, so we'll have our labels like that, and then here's the weird part. For proper format of lab drawings, the title goes underneath, which is counterintuitive to everything you've ever been taught in your whole life. The title always comes first, except here. Okay? The title goes underneath the lab drawing. So this is a specimen from phylum Cestota, which is a type of worm. Okay? It is under 50x magnification, so 50 times magnification, and its approximate size is 1.5 centimeters. Now, you're going to be looking at stuff that's a lot smaller than that, under much higher magnification. So how do I figure out how big something is when I'm looking at it under a microscope? It's obviously too small for me to put a ruler on and go, hey, this thing's about 0.0042 centimeters. No. Okay, you're probably not going to be able to pull that off. So what we have to do is what we call a size estimation. Okay, and it is just that. It's an estimation. This isn't in your notes, so you're going to need to copy it down. Okay, this is how you estimate the size of something under the microscope. When you look through the microscope, you will see a big lighted circle. Okay, that lighted circle is called your field of view. We know how big the field of view is for all three lenses on our microscopes. For our scanning power lens, which magnifies an object 40 times, okay, magnifies it 40 times, our field of view is 4,000 micrometers. Okay, this little symbol here is mu. It's a U with two tails on it. It's actually the Greek letter M. Okay, why do we use that instead of little m for micro? Right, little m already stands for milli. Okay, and we're smaller than that. In fact, we're 1,000 times smaller than a millimeter. Okay, a micrometer is 1,000 times smaller than a millimeter. In other words, the field of view on this lens is 4 millimeters across, okay, or 4,000 micrometers. Okay, so you can use that. If you're putting that in on Google Docs, okay, it's in the symbols section. If you go to insert symbol, you can find mu in there and do the proper insertion of that. All right, I would, if you can't find that, use a U, a small lowercase u, okay, instead, and then I'll know you meant mu. Everybody got that? Don't use m, because that'll be wrong. If you tell me your specimen is 3,000 millimeters, that's 3 of these. Okay? That's kind of big. Not really that big. Okay? A 3-meter amoeba, not going to happen. He could eat you. Okay? That's not good. All right, so make sure you use the right units, because inevitably someone will do that. Okay? They'll put little m, and tell me that I have something that's several hundred millimeters large under a microscope, which would, the only way to be able to do that is if you set the microscope on top of it. Okay? That's the only way it could be under a microscope. Okay? It's too big to fit on it. All right, so our field of view for the scanning power is 4,000 micrometers. When we move up to low power, we increase the magnification. So what we're looking at now is magnified 100 times. But in order to do that, we don't see as much. Okay? What we essentially do is shrink the area we're looking at and make it look bigger. Right? So as you move up in magnification, you see less area, but more detail. Right? It would be like if you were at a, let's say at a football game, okay, and you're trying to find somebody in a bright orange shirt on the other side of the stadium. All right? You wouldn't like go to a high-powered telescope right away to find that person. Agreed? Right? Because how much can you see? How much of the crowd can you see through a high-powered telescope? The person, right? So you got to scan each individual person one at a time through the telescope until you find that person out of 20,000 people. Okay? That's not the most effective way to do it. Most effective way is with the naked eye, find an orange shirt. Then put the telescope on the orange shirt and see if that's the person you are looking for. Okay, everybody follow me there? Find me there. Follow. It's like, where's Waldo? Okay, you're looking for one person. Okay, so you'd first use a wide field of view the scanning power to scan the slide and find your specimen, move it to the middle, and then crank up to the next power of magnification. Okay? If you recall, the other day I told you that there's a pointer, okay, that's inside that field of view. Before you move up, make sure your specimen is on the end of the pointer, so it'll still be there when you move up. Because sometimes when you click to the next magnification, it's so out of focus you can't even tell that anything is still there. You'll know it's still there because you put it on the end of the pointer. All you have to do is focus and it'll be there again. All right? So on low power, our field of view shrinks because our magnification goes up, okay, to 1600 micrometers. No, it's mu. Okay, it's a U with tails. Okay, so it looks like that. Okay, you go into symbol, insert symbol, and it's in there. Or use a U, a lowercase u. Okay. When we move up to high power, again, we go up in magnification a lot. Okay? When you look under high power, you're going to be looking at it 400 times larger than it naturally occurs. 4 times greater than the low power lens. Okay? So it's going to be a lot bigger, and of course, this is going to shrink. Whoop. Okay? Our field of view is going to shrink again. So we'll have a 400x magnification, and our field of view is 4 times smaller than it was on the low power lens. Now it's only 400 micrometers. Why is all that important? Because it allows us to estimate the size of an object. So if I've got my high power lens, I'm looking at something through high power, I know that from here to here is 400 micrometers. And the object I'm looking at is that big. How big is it? Okay, that's not a bad estimate. It's about a quarter, maybe a little less than a quarter of my field of view. I would say I would accept probably anything between 80 and 100 micrometers as an acceptable size estimate for that object. Okay, is that very hard? No. And it is just an estimate, guys. I'm not expecting that you give me like 3 or 4 decimal places after the number. Okay, we're looking at an estimate here. Okay, to the nearest 10 micrometers is probably sufficient. Okay, 80 to 100 micrometers would be the approximate size of that object. All right, everybody with me there? So that has to be included on your lab diagram. Okay, here's the size of the object, this many micrometers. All right, and that's how you do it. It's not a really difficult calculation. Okay, so this is the stuff we just talked about. That's the notes on how to do it. Okay, field of view divided by the number of objects that would fit across will give you the size in micrometers. Okay, that's what we just did there. All right, I would like you guys to answer those 3 questions there, and we'll go through them here in a minute, and then we'll talk a little bit more about tomorrow's lab. All right, let's have a look at these. So, 3 points of the cell theory. Point number 1. Okay, we don't want to say everything here because that would include non-living things. All living organisms are made up of cells. Yeah, I know that seems kind of, anal retentive, and maybe it is, but it's also true. Okay, second point. Right, the cell carries out the basic functions of the organism, and if you recall, I told you that's the most important point. Okay, that's going to come up time and time again in this unit when we start talking about specialization, the role cells play, okay, things like that. Right, and where do cells come from? That's the third point. They come from the division of existing cells. Right? And that's true even for things that don't have a nucleus like red blood cells. Okay? They come from the division of stem cells that are essentially in your bone marrow, liver, okay, places like that where they can be produced. They still come from the division of other kinds of cells. Okay? All right, who are the 4 people considered to be the most important in the development of the cell theory, and what did they do? All right, well, the guy who discovered cells was Hooke. Okay? So Hooke was the guy that discovered cells. That's pretty important. He was looking at cork. It was just a lens experiment for him. Okay. Second guy, first person to look at living organisms, Leeuwenhoek. Okay. And then the two Swedish guys, Schleiden and Schwann, they're the ones who came up with the idea that all things would be made of cells. Okay. They proposed the original cell theory. Okay. Third thing, on what basis can we deduce that plants and animals may have had a common ancestor? Okay, they have a membrane, they have a nucleus, they have a lot of shared structures and processes. Okay, the processes that fuel cells are basically the same. Okay, after you get to glucose. Before you get to glucose, obviously animals and plants are very different. Okay, plants use photosynthesis and animals break down organic material into glucose. Okay, those are two very different processes. Once we have glucose, the fuel for all cells, the processes become identical. All right. All right, so those are the important things there. Now, just back a little bit here, kind of an aside, okay, to this. We talked a little bit yesterday about that, cells come from the division of existing cells, and we talked about how that means that when the moment of conception occurred, that single cell called a zygote, okay, contained everything necessary to make you, okay. As it, of course, divides, okay, these cells become specialized into different groups of cells, okay, through a process that's called embryology. Once the cells begin to specialize, okay, and you have essentially a true embryo, no longer like a zygote, that is where, if you've heard of like stem cell research, those are the embryonic stem cells that some people want to harvest, okay. The benefits of those types of cells is they are destined to become certain types of tissue, right? If you know a lot about embryology and you take an embryo, okay, very early on in its development, you can take groups of cells from that embryo that were destined to become its liver, okay, its heart, its muscles, its nerves, its pancreas, its digestive tract, all of that. You can take the cells from that embryo and culture them, and you will grow, not necessarily a liver, okay, but you would grow the tissue that the liver was made out of, specialized cells that would be part of your liver. All right, does everybody follow me there? Okay, that's the idea behind stem cell research, at least embryonic stem cell research. What is the issue with that type of stem cell research? You would still have to cut out the cancerous cells, right? That's the issue. Is cancer is a problem because it's still your cells, right? And so your body doesn't recognize them as being a foreign invader and essentially takes no action against them. But they grow out of control. And so essentially they end up starving the healthy cells around them as the tumor grows. So you would still have to cut that out. But yes, you could then theoretically replace the section of tissue you'd cut out with tissue that you had grown or cultured from a stem cell, right? Now, what's the problem with that, though? Where do you get the stem cells from? Not necessarily live grown people, but you get them from an embryo. You get them from an unborn baby. And if you take the cells from that embryo that were supposed to be its liver, it's going to die. Okay, that's the issue that we as Catholics have with that type of stem cell research, is that it does result in the death of an embryo. Okay, and that embryo was already at the point where it was starting to specialize cells. Okay, it had all the potential to be a person. Okay, in the eyes of the Catholic Church, personhood, okay, begins at the moment of conception. That's why there's a problem with that type of stem cell research. Now, there are other kinds of stem cells. Other types of stem cells can be taken from, okay, either a grown person, okay, or they can be taken from the placenta after a baby is born, right? My wife and I, we did this after our second son was born. We collected essentially the afterbirth, okay, and it's stored in a like nitrogen frozen area. If any of our children, who both have the same blood type, okay, ever need stem cell type of tissue, we know that we have a match. Okay, it's stored on ice somewhere, okay, and we can use that stuff to grow healthy tissue that would be a match, a donor match for them at any time in the future. Okay, so there's of course, that option that, of course, does not result in the death of an embryo. Okay? There's also what are called pluripotent stem cells, and those can be taken from an adult person. Right? They are harvested either by harvesting bone marrow, okay? Or you can actually have them taken if you have liposuction. Okay? The fatty tissue in your body often contains large amounts of stem cells. Okay? Actually, my knee surgeon is looking at that right now. He's doing research on it. You can have some liposuction done. They collect and concentrate the stem cells in a centrifuge, and a few hours later, they inject them into your knee. And your knee begins to regrow cartilage with your own stem cells, yes. In a different place, yeah. Otherwise, I mean, what's the point of getting the fat sucked out if they're just going to put it back? Okay? But no, they actually, they don't just take, like, they don't take a whole wad of fat and just shoot it into the knee. They actually have to centrifuge it so that the fat cells, which are useless, okay, end up higher up, and the stem cells, which can become anything, end up at the bottom of this tube. Okay? So they would take those out, and then they inject those stem cells into whatever joint you have problems with, and cartilage will regrow. Okay? Now, as you age, of course, that's what causes arthritis, right? Is that your connective tissue, your cartilage wears down, and as you grind on it, it inflames, it swells up, it's painful, it's awful, okay? And you get to be like me and you're old and decrepit, okay? You have no cartilage left, okay? I've had 2 scopes on my right knee. There's no cartilage left, okay? It's just bone on bone now, okay? So it's painful if I don't step quite the right way. Okay, and bone scrapes on bone. That's exquisitely painful. Okay, and so I get like cortisone injections and things like that into my knee periodically. But if I get these cells taken out of my body that can become any kind of cell, all they have to do is be exposed to a medium where they can grow, the inside of my knee, okay? And they'll do that. Okay? Which is pretty cool. It's pretty cutting edge research, but it goes back to this cell theory that all cells come from the division of existing cells, and some of them, if they get the right signal, can become anything. Okay? And that's kind of where that research is headed. It's pretty promising stuff. All right. They've also done some research into the repair of spinal injuries using stem cells. So if you become paralyzed because your spinal column, or your spinal cord, not your column, spinal cord has been severed, okay, they can actually regrow or culture some nerve cells and graft them across the break, and you can regain at least some of your mobility after being paralyzed. Hasn't had any human trials yet, but I saw some research on it that is being done on rats, where they severed the spinal cord of rats, paralyzed them on purpose. I know that sounds really cruel, okay, but rats are disgusting, okay. So they severed the spinal cords of these rats, and then performed this surgery where they cultured nerve cells and grafted them across the break in the spinal cord, and the rats regained a fairly good percentage of their mobility. Okay? Well, yes, but okay. Yeah. They're lab rats. They're grown specifically for that purpose. You go to university and you take introductory biology, that's one of the first things you dissect. Not live. No, they gas them. They use carbon monoxide to kill them. But when they come to you, they're still warm. Yeah. It's a little disturbing, actually, because... no, you're not going to cook the rat. It makes it a lot different to dissect when it's been cooked in the microwave. Okay? No, they don't warm it up. They don't warm it up in the microwave. Okay? Okay. If you go into university, you dissect it. If you take biology, a lot of different things. I dissected, let's see, started with worm, and then squid, and then clam, and then crayfish, fish, so like a perch, rat, okay, dogfish, shark, cat. Yeah. I hate cats. It's like, okay, first thing you got to do is skin this cat. Sweet. No, I hate cats. But no, the cats are the ones that the SPCA has to put down because they're that's those cats. So anyway, yeah, you dissect all those. If you take gross anatomy, which is not gross because it's gross because it's big, you get a human cadaver. Cadaver, a dead person. Yeah. If you take gross anatomy, you get someone who has donated their body to science, and you dissect them. Yeah. No, it's not gross. It's a little weird when you see this person, right? You see, because most of us have not seen a dead body before, right? That's not something most of us experience till you get to that point. Now, the whole process, though, is very respectful. This person has a name, okay? So when you get your cadaver, it's a person, okay? Ours was Lyle, okay? That was his name, okay? They don't tell you his last name, but our guy's name when he was alive was Lyle. Okay, that was the guy we dissected. So yeah, and they take it very seriously. If you joke around at all in that class, like you get tossed. Okay, because this is a person who's dead. Their family misses them. They donated their body to science because they felt that was important. Okay. Well, basically, you look at a lot of things. You look at, like we took the skin off the arm to look at the skeletal muscles of the arm. We also looked at the circulatory system, heart, okay, arteries, things like that, kidneys. I mean, you look at basically all the major organ systems, but on a human. Okay. The closest thing you guys will do in school here is a fetal pig.
Yes, I saw
Yeah,
that.
yeah, you'll do the fetal pig. Yeah. About 20 or 30.